So welcome you all to the next series of our Mendel's laws of inheritance. Today we'll study about the monohybrid cross and the various laws that have been derived from this particular experiment. Okay. Now you know that Mendel did two types of experiment. One was based on a single pair of contrasting character, which we call as monohybrid cross, that we'll study today, and another one was based on two pairs of contrasting characters, which we call as dihybrid cross. Okay. So let us come to the monohybrid cross. In the monohybrid cross, Mendel crossed a true breeding tall plant, okay, with a dwarf plant. True breeding tall plant means it was denoted as homozygous tall, isn't it? Homozygous tall, capital T, capital T. He crossed it with a homozygous dwarf plant. Both of them were true breeding. Got it? So in the F1 generation, what he got? Now he got a heterozygous tall plant. Got it? The plants that he got were all tall in the F1 generation. Now he allowed the F1 plants to self-pollinate to produce F2 generation. In the F2 generation, he got plants in the ratio of 3:1. 3:1. Okay, 3:1. Out of which 3 were tall and one was dwarf. And when we take into account the genetic composition of those plants, he got them in the ratio of 1:2:1. Got it? So this is what he got. Now let us come to the next slide. Here we would be able to know what he found. Let me wipe it out. Okay. Look at it. Now in the first he took tall plant, a homozygous tall. He crossed it with homozygous dwarf. What he got in the F1 generation, he got your the heterozygous tall plant. Now he allowed these heterozygous tall plant to self-pollinate, as a result of which in the F2 generation he got plants in the ratio of 3:1. Okay, 3 were tall and 1 was dwarf. Look at it. Look at this one. This one is capital T, that means tall. Capital T, small t, that means tall, because capital T is the dominant gene. It will be able to express itself. This one is again capital T, small t, so it will again be tall. Now the fourth one is small t in a homozygous condition. So like this one is a dwarf. So he got plants in the ratio of 3:1, where 3 were tall and 1 was dwarf. Now when you take into account the genotypic ratio, you will get the ratio is 1:2:1. 1 was homozygous tall, then these two were heterozygous tall, capital T, small t, and last one was a homozygous dwarf. So he got the ratio of 1:2:1. So what he concluded from this experiment, that character is inherited by factors. Okay, he didn't know about any gene. The term gene was not coined. So he called that character is inherited by factor. Okay. Thus a diploid organism contains a pair of factors. Got it? Capital T, small t, diploid organism contains a pair of factors. A true breeding tall as well as a dwarf contains two similar factors, capital T and small t. Got it? So such offsprings with similar factors for a character are called homozygous. Got it? Similarly, hybrid with different factors is called heterozygous. Capital T, small t, that one was heterozygous, and capital T, small t, homozygous. So basing on the observations of Mendel, Carl Correns formulated principles of heredity, which is now known as Mendel's laws of inheritance. Okay, he formulated principles of heredity known as Mendel's laws of inheritance. Here there is some spelling mistake, single A. Okay, single A. Now next come to the next slide. So Mendel's laws of inheritance, laws of dominance, law of segregation of p-rooted gametes, law of independent assortment, out of which the law of dominance is no longer considered as a law. In the lower classes like +2, law of dominance is taken as a law, but in the higher classes, when we will follow some good books, it is no longer considered as a law. Still then, as per your book, law of dominance, law of segregation or p-rooted gametes, law of independent assortment, these three laws were derived from Mendel's laws of Mendel's experiments. Okay. We will study about law of dominance and law of segregation or p-rooted gametes in this particular video, which is these two are based on monohybrid cross. Now come to the next slide. Law of dominance. What does it say? When two alleles of a particular gene come together in an organism, okay, one that expresses itself is called dominant, whereas the other one is called recessive. So when two alleles of a particular gene, two alleles, okay, now they are present together in an organism. Now the organism will be tall. I have already said in the F1 generation. So here the capital T, which represents the tall character, is being expressed. Got it? So the capital T is the dominant gene. It is able to express itself, but the small t, which is recessive, is unable to express itself. Okay, we have already studied about the dominant and recessive. So dominant gene can express itself in both homozygous condition as well as in heterozygous condition, but recessive gene, small t, can express itself only in homozygous condition. It cannot express in heterozygous condition, because in heterozygous condition only dominant gene will express. Got it? This is what your law of dominance is. Now come to the actual first law of Mendel's, that is law of segregation. It is based on monohybrid cross. And as far as the third law, law of independent assortment is concerned, we will study about it in the next video, which is based on dihybrid cross. Okay. So what does the law of segregation say? It is actually otherwise known as purity of gametes. Got it? Purity of gametes. What does it say? It says that when two alleles of a gene are present together in a diploid individual, two alleles of a gene are present together in a diploid individual, they remain there without being contaminated. Okay, two alleles of a gene are present in a diploid individual without being contaminated. Now, when gametes are formed from the individual, the two alleles separate from each other and pass into the gametes individually. So each gamete receives a single allele and it is pure for that character. Got it? Now when two alleles, they are present together in an individual, gametes are formed from this individual. So each gamete, now the two alleles, capital T, small t, they separate. Okay, and they pass on to the gamete individually. Now each gamete receives a single allele and it is pure for that character. This is what your law of segregation of p-rooted gametes is all about. And this segregation occurs during which stage? During meiosis stage. During meiosis stage. We have already studied about meiosis in the first year, isn't it? So as far as Mendel's experiment is concerned, in the F2 generation of monohybrid cross, out of total 1064 plants studied for stem length, that is tall and dwarf character, 787 were tall and 277 were dwarf. Okay, 787 were tall and 277 were dwarf, with a ratio of 2.84 is to 1. Okay, now is to symbol is not coming. So I think you would be able to understand 2.84 is to 1. That means approximately 3 is to 1. Now when similar monohybrid cross were made by taking into account all the 7 pairs of contrasting characters individually, the segregation of dominant and recessive factor was found out to be the same 3 is to 1. Got it? So from the above, it was concluded, it was found out that pair of parental factors, they separate, and that one factor is present in each gamete, which shows law of segregation. Okay. So this is what your law of segregation is. So today what we studied? We studied about the monohybrid cross, a very easy one, and the law of dominance as well as law of segregation. Okay. Now thank you. In the next video we will discuss about the your dihybrid cross. Okay.